Namaste viewers. Today in this lecture talk, I'll be discussing with you polymerase chain reaction. Okay, let us go through polymerase chain reaction. So what is the full form of this PCR? Polymerase chain reaction. Okay, this polymerase chain reaction mainly running on DNA polymerase enzyme. Okay, and this PCR responsible for the extension of DNA or synthesis of DNA, all right, which adds the deoxyribonucleotides according to the template DNA, right? This polymerase chain reaction can also be known as thermal cycler or molecular Xerox machine. Okay, and this could also you can name as temperature regulating machine. Okay, in this particular polymerase chain reaction, chemical reactions will be happening, right, which proceeds exponential manner. Okay, so this commonly PCR known as polymerase chain reaction. Okay, so you can also ask in the beginning, what is this PCR? Why do we need to study this? Okay, this PCR is a cell-free technique. It means without cell, we are going for the amplification process. Okay, so this PCR is a cell-free technique responsible for the amplification of desired DNA fragment into several copies. Okay, let us go through the introduction part now. In order to study genetic structure and functions, sufficient quantities of DNA of interest must be available for analysis throughout the that particular experiment or particular program, right? For example, in forensic science, if we have a less amount of DNA sample, we cannot check with the different victims. Even while doing research work also, we should have enough amount of the sample, right? Earlier, this was carried out by using microorganisms to generate multiple copies of genes, which is lengthy and laborious process. Okay. Earlier, what we were using amplification of desired DNA fragment, that was by using microorganisms. Now, what we are using, this is the cell-free technique. So, what you can say nowadays, a cell-free technique is available known as polymerase chain reaction, which is nicknamed as People's Choice reaction. Okay. PCR is a technique, okay, in molecular biology to amplify a single or a few copies of segment of DNA to several orders of magnitude, okay, generating thousands to millions of copies of particular DNA sequence. Okay, it means by using this particular PCR machine, we can get from thousands to million copies of particular DNA we can obtain. This techniques as named has made it possible to synthesize large quantities of DNA fragment without cloning it. It means without cloning to any kind of microorganisms or living being, we are obtaining the fragment in a large quantities. Okay, this technology is much easier and requires much smaller quantity of DNA, that is in nanogram, whereas for cloning by using the microorganisms, we need the DNA that is microgram quantities. Okay, if I'm going very quick, you can pause this video and you can go through. Okay, this PCR technique was discovered by Carrie B. Mullis in the year 1985, who worked in Cetus Corporation, a biotech company of United States. PCR was the major scientific development of the year 1989. It is automated, okay, and carried out by specially designed machine. That is nothing but PCR, okay. This PCR is an in vitro. In vitro means that is in the laboratory or maybe in the vials. In vitro DNA amplification technique that allows synthesizing million number of copies of the gene that is a segment of DNA or DNA of interest from a single copy. Okay. Now let us understand little more information about PCR. This PCR can be used to clone a given DNA sequence. Okay, this is DNA sequence in vitro without using living cells during the cloning process. However, this procedure can only be applied when the nucleotide sequence of at least one short segment on each side of the region of interest is known. It means what I'm telling, what are the DNA sample you have? Okay, this is the DNA sample, double stranded DNA, both strands of DNA which are held together by the hydrogen bonds. Okay, if you want to perform this PCR experiment, what you need to do, you must know at least one short DNA segment of each side of the region of interest. It means what are the sequences which are present at this particular end? What are the sequences which are present at this particular end? Okay, the ends of the DNA fragment should be known. For example, I have a very large DNA molecule. Okay, but I want to synthesize this particular region in between. I must know this sequence, okay, and I must know this particular sequence. So same thing written here. This PCR can be applied only when the nucleotide sequence of at least one short, okay, this one short segment on each side, okay, both the side, each side of the region of interest is known, okay. Once we know the each side's sequences, we can design primers, okay. Primers are the single-stranded DNA molecule, okay. These, once we know the sequence, we'll design the primer. These primers which are complementary to the ends of the target DNA, okay. And these primers play a very important role to initiate the extension by the DNA polymerase as DNA synthesis, okay. On next slide, you'll understand better about the primers. PCR producers, PCR producers make use of synthetic oligonucleotides complementary to the known sequences. Polymerase chain reaction may also be defined as a technique in which repeated enzymatic thermal, this is very important, keep in your mind, repeated enzymatic thermal cycles of DNA synthesis and can be carried out to produce large number of specific DNA sequences. Why I mentioned here repeated enzymatic thermal cycles? Because the enzyme, there are what's one cycle, what are the cycle will be 3 steps, one cycle will be there. The 3-step cycle will be repeated many times to obtain many number of copies. Okay, all that repetition of the cycle will be done and extension will be done by the enzyme. So we said here the enzymatic. And why we mentioned here thermal cycles? Because this PCR mixture will be incubated in a series of temperature. Okay, different temperature we are using, a very temperature can be seen. So it is thermal cycles. This technique is so precise and powerful that even a single sequence can be amplified into a detectable amount. This is the specialty about this particular PCR. Now let us understand mechanism of PCR. In order to understand PCR, it is necessary to familiar with the mechanism of in vivo DNA replication. Once we understand the in vivo DNA replication, we can very easily understand in vitro amplification of the DNA through PCR. The DNA replication involves polymerization of nucleotides, right? Using a template DNA strand with the help of oligonucleotides, which acts as a primer strand, right? To which further nucleotide can be added by using the DNA polymerase enzyme. Okay, hope you understood in this particular case. So what I'm going to say here, the DNA application will be that is nothing but the polymerization of nucleotides. What are the DNA will be there, right? That DNA will be replicated into many number of copies. Or polymerization will be happening in the presence of the enzyme, right? With the help of what? Oligonucleotides, which acts as primers. Oligonucleotides means oligo means few nucleotides. Few nucleotides, that is the primers, right? In presence of single stranded primer, this polymerization or synthesis can be seen. Now let us understand the components of this particular PCR. Okay, the DNA is amplified by PCR in an enzyme reaction. Without enzyme, we are not going to perform this particular PCR machine experiment. So we need compulsory enzyme. Which enzyme you want? Okay, you'll come to know. Okay, just keep in mind which enzyme, because usually you'll be asked this particular question in your MCQ. Which this enzyme which undergoes multiple incubations at different temperatures. Okay, each PCR has 4 key components as follows. The first one is template DNA. Template means it is a segment of single stranded DNA. It is known as template. When the double strand DNAs are like this, which are held together by the hydrogen bonds, we can't say this as the template. Okay. So when the hydrogen bonds will break, okay, when the hydrogen bonds are broken here, when we get such kind of strand, we'll say that's as the template DNA, that is single stranded. Then next one is oligonucleotide primers. These oligonucleotide primers are on 20 to 30 nucleotides long, okay, which are single stranded DNA and complementary to the ends, okay, complementary to the ends of the target DNA. And these primers helps in initiation of extension, okay, initiation of extension or synthesis in the presence of DNA polymerase enzyme. Okay, in the presence of DNA polymerase enzyme, this primers will be extended, okay, to obtain the another copy or for the amplification purpose. Right? You are understanding, no? DNA polymerase here cannot initiate on its own on this single stranded DNA. This particular DNA polymerase cannot go and sit on the single strand template DNA. They cannot initiate the synthesis. It means compulsory to this single stranded DNA molecule, they need primers. When primers attaches here, then only DNA polymerase goes there and then binds and synthesis the complementary opposite strands of DNA. Okay, hope you understood now. So to add some more information to this particular primers, there are 2 types of primers. Do you have any idea? Okay. Okay, there are 2 types of primers. That is, one is forward primer. Okay, another one is reverse primer. Okay, next coming to the DNA polymerase. Specifically, which enzyme are we using in this PCR? Yes, good. Which enzyme? Taq DNA polymerase. Okay, Taq DNA polymerase enzyme we are using in this PCR machine to obtain the many number of copies. T, which is the thermus, AQ, that is nothing but the aquaticus. This is the one of the bacillus, which is obtained from the thermus aquaticus bacteria, which are found in hot springs, okay, which can withstand up to 95 to 97 °C. And this polymerase, which has exonuclease activity, okay, and this enzyme responsible for the synthesis of DNA on template DNA and are also responsible for the extension of the primers. What are the primers which goes and attached? No, further these primers extension will be done in the presence of the this particular DNA polymerase enzyme. So this Taq DNA polymerase enzyme, this will synthesize according to the template DNA. According to this particular template DNA, Taq DNA polymerase will keep on synthesizing the new strands of the DNA molecule. Okay. So this DNA polymerase will be adding various DNTPs. Okay. Hope you understood, no? DNA polymerase, which enzyme will be using? Taq DNA polymerase. This enzyme responsible for the amplification of the DNA. So this synthesis repetition can be seen here. That's why I mentioned repeated thermal cycler. Next component is deoxynucleotide triphosphate. In short, we can say DNTP. Somewhere in MCQ or maybe in Viva, you will be asked. Kindly make a note of this one. Deoxynitrogen triphosphates. Okay, then we need react. What are the DNTPs you can use? You can use D-ATP, right? You can use D-GTP, You can use here DCTP, and you can also find DTP, TTP. Deoxyadenosine, deoxyguanine, deoxycytosine, and deoxythymine triphosphate. So all these individual DNTPs you need to put in the reaction mixture. Or what are the PCR mixture tube will be there? No, in this tube you need to provide. You need to put the DNA sample. You need to put the DNA sample. Then primer must be there. Then DNA polymerase must be there. Then many number of DNTPs you need to put. Then all these things will be suspended in the reaction buffer. Okay, this is all about the components of PCR mixture or PCR experiment. Okay, hope you are able to see here. You can see here, this is the single standard primer. Here you can see individual nucleotides, right? And this is the tag DNA polymerase enzyme. As this PCR mainly based on the thermal cycler. Okay, along with that, it is a enzymatic amplification. So the presence of enzyme is very, very important. Then this is the desired DNA molecule in the double standard DNA condition. Okay. Pay attention, kindly be with me. Now let us go through the, what is this template DNA? Template DNA containing target DNA sequence to be amplified. Okay. The template DNA is a complex mixture of many different sequence. Okay. Any DNA molecule can be used as a target sequences. RNA can also be used for PCR by first making cDNA copy using reverse transcriptase enzyme, right? As we already studied in the previous semester, we have the RNA molecule as a genetic material. This RNA molecule will get converted in the presence of reverse transcriptase enzyme. Once this convert this in the presence of reverse transcriptase enzyme, the obtained DNA product will be known as cDNA, that is complementary DNA. Further, this cDNA will be synthesized by the DNA polymerase enzyme to get the pure sample of DNA molecules. Okay, so template DNA means, just keep in your mind, it's a single standard DNA molecule. Now let us go through the oligonucleotide primers. Each PCR required a pair of oligonucleotide primers, right? Whenever we have a DNA molecule which are separated, the primers goes and attached to the opposite strands of the DNA fragment at the edges. These are short single. Okay, this is the oligonucleotide primer and this is the oligonucleotide primer. These are short single stranded DNA molecules, typically 20 to 30 bases, which are obtained by chemical synthesis. Again, in vitro condition, we can synthesize the primers. Okay, you can also have a look about primer designing. Okay, primer sequences are chosen so that they bind by complementary base pairing to be amplified. It means the DNA which you are willing to amplify to that particular ends of the DNA fragments, these primers goes and attach over there. Okay, as I already explained in detail about the primer, right? So there are two types of primers, right? One is the reverse primer, other one is the forward primer. Okay, now let us go through the annealing. Okay, what is this annealing? Like that. Okay, now let us go through your annealing. Okay, this question asked in the examination for 2 marks. So kindly make a note of this one. Annealing, you'll understand later what is this annealing when I explain this mechanism of PCR with the different steps of this particular PCR. Okay, annealing, that is nothing but annealing of the primer to the template acts as a initiator for DNA synthesis. As I told earlier that if anything, a double stranded DNA is there, to this double stranded DNA, DNA polymerase cannot go and attach. So when this DNA polymerase separates, that process of separation known as denaturation. When that separates through breaking the hydrogen bonds, that time what happens? To the single strand DNA molecule, primers go and attach. Okay, annealing or sealing of the primers to the template is known as the annealing. Okay, or attachment. Same thing I have written, annealing of the primer. Primers goes and binds over here, right, to the free ends of the DNA fragment, right? Annealing of the primer to the template acts as an initiator for DNA synthesis. DNA polymerase cannot go and sit on with this particular DNA molecule, double strand DNA molecule. They need primers, or they need, in the beginning, there must be a double strand initiation. Then The tag DNA polymerase goes and binds here, then keep on adding the individual nucleotide according to the template DNA, right? Here also same thing will be happening. Oligonucleotides are, what are oligonucleotides or what are primers? You can write this one, okay? Oligonucleotides are small stretch of desired DNA fragment which acts as primer to synthesize DNA. That is, primers are the initiators of PCR or for the DNA synthesis for the tag DNA polymerase, which are obtained by chemical synthesis. Okay, this you can make a note. Okay, usually during Viva or maybe in MCQ or maybe in 1 mark or 2 mark question they'll ask you. Now let us understand about DNA polymerase. Okay, which DNA polymerase are we using here? Thermus aquaticus. Good. A major improvement in PCR amplification, which came with the discovery of the heat stable DNA polymerase. Whatever we are using right now, the Thermus aquaticus, which is stable to heat. The most commonly used enzyme is Taq DNA polymerase, which is obtained from Thermus aquaticus, a bacterium, okay, that is Archaebacterium growing in hot springs. The polymerase is called Taq polymerase after the genus and species name of the bacterium from which it was isolated. Okay. The Taq DNA polymerase acts best at 72 or maybe 70, one is 72 degrees Celsius or 74 degrees Celsius, and also remain active during heat denaturation step. Okay, maximum temperature more than 90 degrees Celsius or 97 degrees Celsius or 94 degrees Celsius. Up to this temperature also, this Taq DNA polymerase withstand to this high temperature. Okay, in each cycle of amplification. Why in each cycle? As I told, this PCR, which is the repeated thermal cycle, the temperature which will elevate and then decrease, elevate and decrease, this repeated cycles which is going to happening. That time also, this Taq DNA polymerase which will stand to higher temperature. Okay. The role of the Taq DNA polymerase in PCR is to copy DNA molecules, right? Their job is to copy DNA molecules like a Xerox machine shop. The enzyme binds to double-stranded DNA and synthesizes a new strands which are complementary to the original strand. For example, if this is the double-stranded DNA molecule, here the hydrogen bonds will be broken down. Once hydrogen bonds are broken down with the higher temperature, they'll become single-stranded DNA. Okay, when they become single-stranded DNA, primers go and attach to the free ends of the DNA fragments to the template DNA. Next, what happens? Enzymes binds. Okay, here also enzymes goes and binds here. Okay, here one primer will be known as forward primer. I told you there are two types of primer. So this will be your forward primer. Okay, and this will be reverse primer. But the enzyme binds to double-stranded initiation DNA region and synthesize a new strands. Okay, here that will synthesize new strands according to the template DNA. Okay, or complementary to the original strand. This is the original strand. Okay, and here this is the original strand. Based on the original strands, new DNA strands are going to be synthesized. The DNA polymerase requires short region of double-stranded DNA to get started. That is nothing but the primers, right? That is nothing but the oligonucleotide primers. In PCR, this is provided by the oligonucleotide primers, which creates short double-stranded regions by binding on either sides of the DNA strands to be amplified. In this way, the primers direct the DNA polymerase to copy only the target DNA sequence. Now let us understand about DNTPs. Deoxynucleotide triphosphate molecules corresponds to the 4 bases present in DNA, that is adenine, guanine, thymine, and cytosine, which are substrates for DNA polymerase. If we have the DNA strand, okay, in the reaction mixture, need to provide the DNA strand also, DNTPs need to provide, then reaction mixture, right? Then primers you need to provide, and Taq DNA polymerase you need to provide. If the single stranded and primers goes and attach over there, then Taq DNA polymerase also goes and attach. For this Taq DNA polymerase enzyme, individual nucleotides are the substrate. If there is no individual nucleotides, okay, provided in the in vitro condition, as the cell-free technique, we need to provide everything. Then what happens? There is no further DNA synthesis. So for this tag DNA polymerase, whatever you provide individual DNTPs, no, that is going to act like a substrate for the enzyme, which adds the individual DNTPs according to the template strand. Each PCR requires 4 DNTP, that is DATP, DCTP, DTP, that is deoxyguanosine triphosphate, which are used by the tag DNA polymerase as building blocks to synthesize new DNA or for the extension of DNA according to the template DNA. Okay. Now let us understand here further more information about PCR. PCR allows the amplification of target DNA, right? Sequence through repeated cycles of DNA synthesis, right? Each molecule of target DNA synthesized acts as template for the synthesis of the new target molecules in the next cycle. As a result, the amount of target DNA increases with each cycle by using the reaction components. Okay. Now let us understand what are all these things through this particular following steps. Each cycle of DNA synthesis involves 3 stages or steps. First one is denaturation. Okay. Denaturation in the sense whenever the DNA strand which are held together by the double stranded DNA molecule, no, when we increase the temperature, both the strands will get separated. Okay. So denaturation means separation of the strands of DNA fragment into single stranded. Then primer annealing. Primer annealing means once the DNA molecules are separated like single strand, the primers goes and binds to the free ends of the DNA fragment. Ends of the DNA fragment. The next one is polymer extension. Once this primers goes and attached to the ends of the template DNA, primers goes and binds over there, right? And once this tag DNA polymerase enzyme binds, it will keep on adding the complementary nucleotides according to the template DNA. Okay, according to template DNA, it will keep on adding here. And here also simultaneously, it will keep on adding. And you can see the extension here, right? You can see the extension. Okay. So from 2 strands, we got here the 4 strands. So this is all about the PCR. So PCR mainly works on 3 steps: denaturation, primer annealing, and the polymerase extension. So these are the requirements of this particular PCR experiment. Target DNA, which contains the sequence to be amplified. Then 2 oligonucleotide primers, which binds to the 3' hydroxyl of both strands. Okay. Then DNTPs, that is deoxynucleotide triphosphate, which is like a DNA building units. And one more thing you can write it in the primers is that you can write reverse primer and the forward primer. Then thermostable tag DNA polymerase, okay, enzyme that catalyzes the reaction. Magnesium ions as a cofactor of the polymerase enzyme. Then buffer solution, which maintains pH and ionic strength of the reaction solution, which are suitable for the activity of the enzyme. So these are some of the major requirements. Kindly go through this one, and you should compulsory write all these 6 relevant requirements in your answer. Now let us understand here what is denaturation. Okay, denaturation, that is the this denaturation is brought by the heat condition. So you can say heat denatured or melting of target DNA. It means the mixture is heated up to 94 degrees Celsius. At the temperature, what happens? What are the double stranded DNA molecule will be there, which are held together by the hydrogen bonds, right? When you increase the temperature in the PCR machine to 94 degrees Celsius, at this temperature, what happens? This hydrogen bonds that holds together the 2 strands of DNA molecules are broken, okay, are broken, causing the molecule to denature. Once they are broken, then they'll become like single stranded. So this is nothing but the denaturation, very simple. Okay, kindly be with me. Hope you understood this denaturation. No? Denaturation, that is nothing but the separation of DNA strands. Okay, that is at the temperature 94 degrees Celsius. In some of the notebook, if they mention 95 or 93 like that or 97, don't worry. Any one particular temperature you write. because the PCR machine, which differs from one manufacturer to other manufacturer, any one particular figure you can write. And this denaturation will be happening within how many minutes? This denaturation will be happening within 1 minute. Okay. Now let us go to the primer annealing. I have told you already in the previous slides what are these primer. Okay. This mixture is cooled down. So in the previous condition denaturation, what was the temperature we kept there? 94 degrees Celsius temperature was there. Now this particular PCR mixture, which is cooled down to or lower to 50 to 60 degree Celsius. Okay, we are reducing from 94. We are reducing here from 94 degrees Celsius to how much? 50 to 60 degree Celsius. Okay, some of the book they specifically they'll mention 55 like that. Okay, you can write that also. You can write 50 to 60. A temperature. Okay, this temperature allows the binding of the primers to the single stranded DNA without permitting the double helix to reform between the template strands. This process known as annealing. Okay, here primers are the initiations of PCR that anneal to the DNA molecules at specific position. For example, we found after the denaturation, we got this kind of template DNA, right? When we subject to 94 degrees Celsius, we got this kind of template DNA single stranded. When we reduced the temperature to 50 to 60 degrees Celsius, at that time primers goes and binds to the free ends of the DNA fragment. Okay, this is nothing but the annealing, and this particular region is the initiation region for the DNA polymerase. Okay, to obtain the specific products, we need this kind of double stranded initiations along with the primer molecule. Now let us understand extension or elongation or polymerase extension or synthesis. This stage is carried out at the temperature at which DNA polymerase enzyme is most active. The temperature is raised to 72 degrees Celsius. Okay, denaturation was at the 94 degrees Celsius, right? And then primer annealing, right? That is was at the 50 to 60 degree Celsius. We reduced, and now the temperature will be again increased to here 72 degree Celsius. See, we are going through series of temperature, right? Incubation. So this will be your denaturation, this will be primer annealing, and this will be extension, right? In this particular stage, carried out the temperature which DNA polymerase is most active. It means when we increase the temperature in the PCR machine to 72 degrees Celsius, okay, that time DNA polymerase works best. It means this is the optimum working temperature for the tagged DNA polymerase that is present in the reaction mixture. Okay, here the tagged DNA polymerase attaches to the ends of each primer, okay, and synthesizes new strands of DNA. Okay, primers which are already attached over here. Once the primers attached, tagged DNA polymerase goes and binds over here. And here also tagged DNA polymerase goes and binds, right? And start synthesizing new strands of DNA according to the template DNA, right? According to the template DNA. Now we have from 2 strands of the DNA, we have 4 strands of DNA. This completes one cycle of PCR. Okay, this is the one cycle of PCR. One cycle of PCR means denaturation, primer annealing, and extension. Okay, this is the one cycle. The same cycle will repeat again. Okay, extension, then denaturation, then primer annealing, then extension. Then again denaturation, then primer annealing, extension. That's why this process is known as repeated thermal cycler. It means whatever from the 2 strands we obtained here the 4 strands, right? And these 4 strands again will go to the denaturation. This strand will go to the denaturation, and this strand will also go to denaturation. Same cycle repeats. Okay, denaturation, primer annealing, and the synthesis or extension. The temperature is increased back to 94 degrees Celsius. Okay, the double stranded DNA molecules, each of which consists of strands of the original DNA, and one set new strands of DNA again denature into single strand. This begins a second cycle of denaturation, annealing, and synthesis. At each and every cycle, there are 8 DNA strands which are obtained. Okay. By repeating the cycle 30 times, the double-stranded DNA molecule is converted into 2 billion new standard DNA molecules can be seen. Annealing, that is nothing but, I've told you in the previous slide, the pairing of complementary single strands of DNA to form double helix in PCR. So now let us go through what is this primer. Primer, which is a short sequence of oligonucleotides that hybridize with the template strand and provides initiation for the DNA synthesis. Make a note of this one, but 1 mark or 2 marks usually you'll get. Now let us understand in brief, in single slide, what is this denaturation, primer annealing, and elongation. Okay, what I have given in this particular slide, this is enough information. You can elaborate this. Okay, a PCR cycle consists of 3 steps. Okay, first one, what is that? Denaturation. Okay, here you can see the denaturation, then primer annealing and elongation or extension. Step 1, denaturation. Reaction mixture is heated to 94 degrees Celsius for 1 minute. Okay, the temperature denatures. What is the temperature we have increased over there? That temperature which increases the DNA sample to 94 degree Celsius. Okay, at that temperature, 2 strands of DNA fragments which separates or break. You can see the breaking of hydrogen bonds between them, and we obtain the single stranded DNA fragment. So earlier there was the double stranded DNA molecule here, right? When this double strand DNA were subjected to this PCR mixture under 94 degrees Celsius, we obtain the single stranded DNA fragments, right? Where hydrogen bonds are broken down. Next, step 2, primer annealing. Once this kind of separated segments you got here, when you reduce the temperature to 55 degrees Celsius or 50 to 60 degrees Celsius for 1 minute, at that particular temperature, primers goes and attach. Here I have showed one particular set. At a time, many this particular process will be happening with the other DNA molecules. Right, 1 minute. And primer binds to the 3' end of the target DNA at both stands of primer. That is nothing but primer annealing. For example, I said here, this particular primers which goes and binds to the 3' end of the target DNA, right? 3' end of the target DNA means here, whenever you take this any kind of DNA molecule, one end will be having 3'. Okay, let us write this as 3'. Okay, and this as 5'. And the opposite strand, this as 3' and this as the 5'. Always the primers which will be attached to the 3'. If this is the 3' and this will be your 5' and this will be your 3'. Okay. This condition, what happened? This will be known as the forward primer. Okay. This will be known as forward primer and this will be known as the reverse primer. Here 3' means opposite strands. You are going to get the 5'. Okay. And this will be your 3'. Okay. Denaturation for how many minutes? Here denaturation, which is happening within the 1 minute, primer annealing within 1 minute. Now let us understand elongation or synthesis by the thermostable DNA polymerase, which is obtained from the Thermus aquaticus organism. Okay, this tag DNA polymerase works best at optimum temperature is 72 degrees Celsius. Okay, for 2 minutes, this 72 degree temperature will be maintained. At that temperature, tag DNA polymerase enzyme, which goes and binds to this particular primer initiation region. Okay, primer initiation region that goes and binds here. Once that goes and binds, this tag DNA polymerase, which will keep on adding the individual nucleotides according to the template DNA. Okay, then hydrogen bonds can also be seen over here. Similar, you can also see here, as and when this tag DNA polymerase moves, it will read this DNA, which are nitrogenous bases, which are present here, and keep on adding the individual complementary DNTPs according to the template DNA molecule. Okay, that will be within how many minutes? 2 minutes. Duration to complete a cycle is 4 to 5 minutes. Okay. In each cycle, number of DNAs get doubled. Earlier it was 2. See here. Earlier we obtained here the 2 strands of DNA fragment. After the synthesis, how many strands we are getting? We have got here the 2 strands, right? Here 1 strand, here 1 strand, 2, 3, and then 4. Okay. So every time DNA gets doubled. From the 2 strands, you'll get 4 strands. From 4 strands, you'll get 8 strands. Then 16 strands. That's why exponentially we are getting. Okay, that's why we'll say here this PCR is a chemical reactions which proceeds in an exponential manner. Okay, so how many minutes we have taken for this one particular cycle? The step 1 denaturation, step 2 primer annealing, and step 3 elongation. All this will be happening within the one cycle. Okay, one cycle is this denaturation, primer annealing, and the elongation. In this case, what happens? The second step, same reaction which repeats. Whatever this DNA stands, we have here, no, the double stranded DNA molecule here, double strand. This double stranded DNA again, which goes to the denaturation, okay, annealing, and then extension. And this strand also, which will be going through the process, denaturation, annealing, and the extension. Further, when we do here, how many strands will get? Here 4 strand will get further, and here 4 strand will get further. So from 2 to 4, from 4 to 8, sorry. Yeah, 4. You are going to get the 8 strands from 8. From this 4 again further will be going for the another cycle. 4 will go to another cycle, will get 16. Like that you are going to get in the exponential manner in the particular amplification of DNA. You can keep this particular graph also. Okay, this is the graph, this is the time, okay, in minutes, and this is the temperature. It means in the previous that how many minutes we require for one cycle. Okay, this is 1 minute, right? This is 1 minute, and this is other 1 minute, and this is 2 minutes. So it means one cycle requires around 4 minutes. How many number of sample you want, how many number of copies you want, like that you can adjust the instrument for minutes. Okay, in one cycle, this particular instrument what we are using, that requires 4 minutes. So how many number of copies you want, like that you need to adjust the time, and also need to provide the all requirements. So here I've written this is the time, okay, and here I've written temperature 50, 60, 70, 80, 90, like that. So within 1 minute, in this particular 1 minute, what happens in the PCR mixture? Denaturation will be happening within the 1 minute. That will be for how, what's the temperature? 94°C, 94 or 95, like that. Okay. Once denaturation has happened, then the temperature is reduced or cooled down to 50 to 60°C. Or 50 to 60 means you can also write it over here, 55°C. That time what happens? The substrated DNA strand, which are template are there, no, to that primers goes and attach. That is nothing but the annealing that will be happening within 1 minute. Again, temperature will be elevated to 72°C, which is the best optimum temperature for your DAC DNA polymerase enzyme. Here, DAC DNA polymerase goes and binds to the double strand initiation region of the primer, and further primer extension can be seen according to the template DNA. Okay, this will be within 2 minutes. So this is the first denaturation 1 minute, annealing another 1 minute, extension requires around 2 minutes. So one cycle we are going to complete within 4 minutes. Okay, further what happens? Again, further this instrument will increase the temperature to 94°C. After 1 minute, again this will be reduced to 55°C, and next further will be increased to 72°C. Okay, and 72°C, and goes on like this. Okay, this is about the polymerase chain reaction. Next, let us go through, this is the PCR machine. In the PCR machine, you can use target DNA, as you can take plasmid DNA or genomic DNA, or you can take the PCR product or any annealed oligonucleotides, or even cDNA. cDNA means the DNA which is obtained from the RNA molecules. From those DNA, from those RNA, if you have converted in presence of reverse transcriptase enzyme, no, those cDNA can also be used in the PCR machine. So once DNA, after the denaturation, you are going to get the single stranded template DNA. To this single strand template DNA, primers will go and attach. You can see this is the reverse primer, right? Always primers goes attached to the 3' end. So this will be moving from in this direction, 5' to 3', which is reverse primer, and this is the forward primer, which moves from here this condition. Okay. Then amplification, first cycle, you're going to get the 4 copies, second cycle, 8 copies, so on and so forth. You are going to get first cycle 4 copies, second cycle 8 copies, third cycle 16 copies, fourth cycle 32 copies. At 30th cycle, you are going to get the 2 billion copies of the sample. If you consider this as the DNA, you can see the DNA here, okay? And this is the primer which is attached, and this is the Taq DNA polymerase enzyme, which is keep on adding the individual ribonucleotides according to the template DNA molecule. Okay, so in the reaction mixture, you need primer, Taq DNA polymerase, nucleotides, and DNA you require. Now let us go to the components. DNA sample, that is a target DNA, then primers, nucleotides, Taq DNA polymerase, mixing buffer, a reaction mixture, then PCR tube. We require. In the reaction mixture, what happens? First, denaturation happens at 94 or 95 degrees Celsius. What happens here? Stand separate. Very simple diagrammatic representation I have given here. Kindly make a note of this one. Next, when we reduce the temperature to 55 degrees Celsius, primers bind to the template. That process is known as annealing, forward primer. This is the forward primer, and this is the reverse primers goes and attach. One at the temperature 55. When the temperature is again increased to 72 degrees Celsius, we can see this particular Taq DNA polymerase goes and attach here and start synthesizing the individual nucleotides according to the template DNA. And here we are getting the new product. These two are the new product, right? And these are the original. So each time the original DNA fragments will separate and which go through the repeated cycle. So from the 2 strands, we got this. Here you can see the 2 strands here, right? From this 2 strand, we are getting here the 4 strands. Like that, again, here what happened? This particular strand again, which goes to the denaturation, and this also goes to denaturation, annealing, and extension. Denaturation, annealing, extension, like that repeated thermal cycle can be seen. Here also you can make out very clearly. Here in this diagram, I got here 98. You can make a note, correct this one, 98, 94, or 95. You can make a note. Okay, this is the region of interest, 5' to 3', and this is 3' to 5'. Next, what happened? Denaturation, where temperature is increased to separate DNA strands at 94 degrees Celsius. The hydrogen bonds are broken down here. Then when the temperature is reduced to 55 degrees Celsius, the primer annealing can be seen here, right? What we have done here, temperature is decreased to allow primers to attach to the base of the template DNA molecule, or these primers base pair to complementary DNA template. So this is the template DNA and this is the primer. Okay, here you can see this as the forward primer. Okay, and this is the reverse primer. Again, temperature will be increased to 68 to 72 degrees Celsius for the extension. Here in this case, what happens? Polymerase enzyme, which goes and binds to this double stranded initiation region, okay, on both the side. And here polymerase extends primer to form nascent DNA strand, okay, which will attach here. And from the 2 strands, you obtain here the 4 strands. One will move this side, other will move this particular side. So the newly synthesized strands can also be known as nascent DNA strands. You can see here all this cycle which is happening within 4 minutes. Why? Because denaturation 1 minute, annealing 1 minute, and extension 2 minutes. So within 4 minutes, we are going to finish one particular cycle of the PCR. So you can make out first cycle, second cycle, third cycle, and here fourth cycle. See, original sample was only this one. Blue color was the original sample. Okay. At the 4th cycle, you are going to get the 32 copies. If we going further for the 30th cycle, no, that time we are going to get the 2 billion copies. It means we can see here exponential amplification. Okay, it means here the process is repeated and the region of interest is amplified exponentially. So this is all about the PCR machine and PCR mechanism. Okay. So now let us go through the brief information about the PCR. PCR is a technique that enables the multiple copies of DNA molecules, which is generated by enzymatic amplification of target DNA. Hope you now understood why we are mentioning here enzymatic, because without enzymes, DNA molecule cannot be amplified. So you can say enzymatic. As temperature, we are increasing in a series. So you can also say this particular PCR as thermal cycler. Okay. It is a piece of equipment that makes the PCR mixture to be incubated in a series of temperature and that are varied from pre-programmed manner. Okay. And this PCR which goes through the three stages or three steps. Okay, denaturation at 92 degrees Celsius or 94 degrees Celsius for 1 minute, annealing 52 or 54 or 55 degrees Celsius for 1 minute, and extension 72 or 74 degrees Celsius for 2 minutes. Okay, we can give this particular introduction part related to the PCR, and this was given by Carey Mullis in the year 1985. Then how to study PCR product? To study this PCR products, amplification of product in a various methods we can apply here. Okay, there are so many ways are there not to gain information about DNA molecule that acts as original template. A wide range of procedure have been devised for studying PCR products, in which there are three techniques are particularly important, that is gel electrophoresis or PCR products. What are the PCR products we obtain now? We can go through the electrophoresis process or cloning of PCR product. Again, you can introduce these PCR products to the bacterial host system through the plasmid DNA and you can check. Okay, then sequencing of PCR products. To understand the sequence of nitrogenous bases, you can also go through the sequencing of the PCR products. When we go for sequencing, we'll say as DNA sequencing, we have two types of methods. Okay, one is the Sanger dideoxy method of DNA sequencing, other one is the Maxima Gilbert method of DNA sequencing. In that, any one method we can use for the sequencing of PCR product. What do you mean by sequencing? Study of alignment of the nitrogenous bases is known as DNA sequence or sequencing. Okay. Now let us go through the limitations of this particular PCR. Most polymerase enzymes are able to rectify errors by reversing over the mistakes and resynthesizing the correct sequence. Okay, and that is known as proofreading. Okay, once our DNA polymerase in the in vivo condition that moves from 5' to 3' direction, that will move back again from 3' to 5' region, and that will rectify the mistakes and resynthesize the correct sequence. That is known as proofreading. That kind of proofreading is not there in this particular Taq DNA polymerase. Okay, so proofreading function depends upon the polymerase. Some may have or some may not have. Taq DNA polymerase appears to lack proofreading activity and as a result unable to correct its errors. This means that DNA synthesis by the Taq DNA polymerase is not always an accurate copy of the template molecule. Okay, but the error rate has been estimated at one mistake for every 9,000 nucleotides DNA that synthesis. Okay, so amplified DNA should be studied directly rather than being cloned to the organism. Okay, now let us understand here the applications of PCR in brief. New applications are created every day. Every day is keep on changing. Let me list some of the applications. For example, to amplify DNA fragments isolated from organisms. Then to propagate DNA for gene manipulation and construction of DNA libraries. And this PCR is also used in sex determination of embryo. And even also you can see in the forensic science in DNA fingerprinting. Okay, then PCR products can be used for mapping genes. Okay, PCR products can be used as probes. Probes means are the single stranded known fragments. Okay, and this PCR can also be used to detect genetic diseases. Okay, PCR can be used to identify genotypes. Then PCR can be used to sequence DNA directly. Okay, these are the some of the applications. You can make a note of this one. Even further, if you want to go through, let us go through these applications also again. PCR is used extensively as a research tool, which has greatly improved our ability to study genes. Okay, most studies in molecular genetics involve use of PCR. For example, DNA amplified PCR can be used for DNA sequencing as a probe in northern and southern blotting to generate clones. PCR amplification can be seen in the medicine as well as in anthropology and archaeology. Okay, it is also an important tool in the biotechnology industries. If I'm going very quick, kindly pause this video and make a note of this one. Any three applications enough for your examination point of view. Okay. PCR has made important contributions in many areas, which include inherited diseases, cancer research, forensic science, prenatal diagnosis using PCR. Okay. Further, a study of DNA polymorphism using PCR, whereas in this particular case, human genetics using PCR, recombination data, PCR in gene therapy, PCR analysis of plant viruses, detecting mutations, molecular mapping, PCR preparation of molecular markers, then PCR for research, etc. We can go through. And nowadays, it is a trend that we are using different types of PCR to detect the this particular COVID virus. No, so there will be using RT PCR. We have different types of PCR. For example, we have real-time PCR, right? And this is the traditional PCR we have, right? And we have the RT-PCR. Okay, this RT-PCR we are using in the laboratory to find the whether the person is got infected with this COVID virus or not. So usually in COVID-19, which is caused by SARS-2, that is severe acute respiratory syndrome, coronavirus, where that carries genetic material as RNA molecule. Okay, which is the nowadays become pandemic, right? So to detect whether the person is suffering from this particular COVID-19 SARS or not, first what they'll do, they'll take the swab. Okay, they'll take this swab from the throat, and nasopharyngeal swab will be collected, right? And these swabs will be collected from in the sample collector, and this collector sticks, which is called a VTM. Okay, the collected sample will be transferred into the VTM, that is viral transport medium. Okay, then that will be transported to the laboratory. How they'll take the sample? By using the long stick that will be like a earbuds, like that. Through that, by using that, they'll be taking the sample. In laboratory, the collected VTM, that is viral transport medium, will be subjected to nucleic acid extraction. Okay, they what they'll do, extraction of RNA with the various chemicals they'll be doing, and later that will be subjected to the RT-PCR. RT-PCR is nothing but what? Reverse transcription of PCR they are going to subject. Okay, in this particular PCR, what happens? Why we name this as RT-PCR? Because here COVID virus genetic material is the RNA molecule. Okay, that genetic material RNA molecule will be get converted into cDNA molecule, right? In the presence of reverse transcriptase enzyme. Once this RNA molecule is converted into cDNA molecule, further this cDNA molecule will be subjected to the conventional PCR, what I explained in the earlier one. Okay, so RT-PCR is different, then conventional PCR is different, and real-time PCR is different. Okay, if time permits, in next class I'll do the lecture talk on RT-PCR and real-time PCR. Whatever explained right now, in the previous slide, that is about the conventional PCR. Okay. Hope you understood today's explanation on the polymerase chain reaction, components, mechanism, and the process of that particular PCR. Okay. I, Dr. Pratibha K, signing off for today. Stay tuned with me. I'll be back with some more videos. Thank you for watching this video, and I request everybody kindly subscribe this channel to support and also share to your friends. Thank you.